Hello, everybody. My name is Álvaro López García from the Spanish National Research Council. I'm the project coordinator of the EOSC SIESTA project, and it is my pleasure today to present this webinar on sensitive data anonymization, data privacy tools, and trusted repositories that we organized together with our colleagues from the FIDELIS project. Our workshop today is organized in 4 different sessions where we will cover the tools that we have developed within the SIESTA project that relate to data privacy, metadata privacy assessment, and synthetic data generation, together with an overview of the trustworthy data repositories that is an evolving field in the EOSC ecosystem. Firstly, in our first session today, we will cover 2 general purpose tools, ANHANA and PyCanon, that can be used to anonymize tabular data using the most widely known anonymization techniques. Then we will move to 2 more specific tools in the field of neuroimaging. On the one hand, we have MetaPrevits, focused on anonymizing metadata of neuroimaging datasets, and then BitScramble, a tool that allows neuroscientists to generate a randomized version of an existing bits dataset that will preserve the structure of the data, but without sharing any identifiable information. Then, to conclude, we will explore the ecosystem of secure data repositories for data publication in the European Open Science Cloud ecosystem, together with our colleagues from the EOSC FIDELIS project. Please also remember that you have this Slido page available. This is also available in the YouTube chat link in order to ask questions during the webinar that will be posed to the speakers at the end of their talk. With this brief introduction, I would like to pass the floor to our first speaker, which is Judith Sainz Pardo. Judith is a doctor in science and technology from the University of Cantabria. She's specialized in privacy-preserving techniques in data science environments and has been working in several European projects like EI for EOS and SIESTA in this regard. She is a researcher at the Advanced Computing and E-Science Group of the Institute of Physics of Cantabria, and she has developed the Python libraries ANHANA and PyCanon and TrustGoodDP that will be explained in the following session. So I don't want to have any more time. So Judith, the floor is yours.
Thank you very much, Álvaro, for the introduction. I hope that you can see my slides now.
Yes, we can see your slides.
Thank you. So thank you very much again to all of you that are attending to this webinar. And as already mentioned, I'm going to start talking a little bit about data anonymization tools. We are going to focus on 2 tools that are mainly for anonymizing sensitive tabular data, but also for checking the level of anonymity of a dataset. But first, as I have already said, we are thinking on tabular datasets. So let's start with some basic concepts that we need to know in order to understand how do these tools work. So first, when we are dealing with a tabular database in which we may have sensitive information, we have the identifiers. And the identifiers are the features, the columns in the database that make it possible to identify an individual unequivocally. And of course, some examples can be the identification number, the phone number, the email, but also the name and the surname. The identifiers allow us to, of course, identify unequivocally an individual, but then we have the quasi-identifiers that are the features that, when combined, allow us to identify someone in the database or a group in the database. And such information can be more accessible to an attacker than, for example, the ID number. And we can think about the age, the date of birth, the city of residence, or the gender. Such features are known for, could be known for an attacker, and then when we combine them, we could be able to infer who is the individual in the database with such features. Then if we are dealing with sensitive data, of course, in our database, we may be dealing with sensitive attributes that are the columns in the data set that should not be associated with an individual because it's the information that we want to protect. Imagine a census database. One sensitive attribute could be the salary, or we can imagine the database from a hospital. Then we may want to protect the illness of the patient, and that will be the sensitive attribute. And then when we have our data set, we can define, well, we can have different equivalence classes, which are the partitions of the data set in which all the quasi-identifiers are equal concerning, well, all the quasi-identifiers have the same values. And it allows us to have that if one individual is in a 13 equivalence class, there will be at least other 13 number of rows that are equal concerning the quasi-identifiers. So all the records that are in a same equivalence class are indistinguishable concerning such features, the quasi-identifiers. And in order to anonymize the data, we need to define different hierarchies. And the hierarchies are the transformations that we apply to a certain column on our data set in order to generalize it. And we can think, for example, on the age and how we can anonymize, how we can generalize the age. We can define different intervals of length 5, 10, whatever. And that's, we will be defining different level of hierarchies. And this concerns the basic concepts on our tabular dataset. So now, what are the tools that we can apply in order to anonymize tabular databases? Let's start with the most simple one, and which is widely used, but is extremely simple, which is canonymity. And we have that a database verifies canonymity if for each equivalence class we have at least car rows, which means that if we are an individual that belongs to a certain equivalence class, then there will be at least K-1 individuals that are equal to us concerning the quasi-identifiers. And we can see this table here in which we have that this database is K-anonymous because if we have the quasi-identifiers age, sex, and city, we have one equivalence class with length number 2, and then we have a second equivalence class with length 3. So we take the minimum, and this database will be K-anonymous for K equal to 2. Of course, this is quite simple in this quite a small example, but we will need to be able to scale this to, of course, more complex databases. And here we can also find one problem. If we are one individual in this first equivalence class, even if we don't know if we are the first or the second individual in this equivalence class, it will be immediate to infer the sensitive attribute, which will be the disease. It will be immediate to know that these individuals have pneumonia. In the second equivalence class, we have 3 options for the disease, but for the first equivalence class, it's immediate to know that the individuals have pneumonia. And in that regard, we introduce cell diversity because we are going to focus on the diversity of the sensitive attribute. So we say that a database verifies L diversity if in each equivalence class we have at least L different values of the sensitive attribute. Here in this equivalence class, L will be equal to 1, so we are not protecting at all the diversity of the sensitive attribute. And here in this definition, I say that this is only for one sensitive attribute, but in some cases, we may be dealing with multiple sensitive attributes. For example, in this database, we have the disease as the sensitive attribute, but we may also have other sensitive attributes, such as the last cause of the hospitalization of the patients, whatever. So how can we deal with multiple sensitive attributes? We can think in 2 options. In the first one, we will harmonize the quasi-identifiers with mean, which means that if we have multiple sensitive attributes, we will apply, for example, L diversity for each sensitive attribute. We will apply the other techniques that I mentioned and that I will mention in the following, and we will keep the more restrictive value for all the sensitive attributes. For example, for L diversity, we will apply L diversity with a certain value of L for all the sensitive attributes, and then the value of the global value of L diversity for the dataset will be the minimum value of L, or the largest value for alpha in the case of alpha canonymity that I will explain in the following. However, in some cases, some of the sensitive attributes could act even as quasi-identifiers. Imagine a case in which one of the sensitive attributes that we have can be known by an attacker and can be useful to infer information about other sensitive attributes. We can think about a database in a hospital in which some of the sensitive attributes can be known for an attacker. Imagine that it is a database in which we have patients with cancer. One sensitive attribute could be the cancer type. Another sensitive attribute can be the stage. So one sensitive attribute can be useful for inferring information regarding the other. In that case, what we are going to do is to update the list of quasi-identifiers depending on the sensitive attribute that we are anonymizing or analyzing. So if we are analyzing the sensitive attribute concerning the type of cancer, we will need to add to the list of quasi-identifiers the remaining sensitive attributes that can act as quasi-identifiers. So then we have already seen two definitions for two anonymization techniques that are widely used in the practice. And concerning L diversity, there are other versions that we can consider. I'm not going to stop into the definition details, into the details of the definition, but we can consider the entropy version of L diversity. We can even consider a recursive version that is even more strict than the previous one. And we can also consider alpha anonymity, which has the same, let's say, idea that we have with L diversity, but in this case, we'll have our canonymous database, and we want to verify that the frequency of each value of the sensitive attribute is not greater than alpha in its equivalence class, in order to have, let's say, a measure of control of the frequency of the value. So, for example, here, in this initial database that we have, the value of alpha will be 1 because the distribution of this value in this first equivalence class of this attribute pneumonia in this equivalence class is 1. So we will not have diversity. It's another measure quite similar to L diversity. On the other hand, we may be interested in measuring the distance between the distribution of the sensitive attribute in the whole database and the distribution in an equivalence class in order to find differences between the global dataset and specific groups, because that may allow an attacker to infer more information. In order to do so, we can define T-closeness, and T-closeness measures such distance between the distribution of the sensitive attribute in the original database and the distribution in the equivalence class. And in order to measure such distance, we can use different distance metrics, such as the equal distance for the case of categorical attributes, or the Hermover distance, for example, for the case of numerical ones. And we can also measure such, let's say, distance between the distributions with other metrics, such as delta disclosure privacy, which calculates the probability of a certain value of the sensitive attribute in an equivalence class and the same, but in the whole database, and we have it bounded. We have a lower and an upper bound given by delta and minus delta. And finally, same idea, but with the notions of beta likeness. We have the basic notion and the enhanced one, in which we are again interested in measuring, in this case with the relative distance function, the distance between the distribution of a sensitive attribute in an equivalence class and that in the whole database. And we can consider this basic notion in which we have this distance, the maximum distance bounded by beta, or we can also consider a more restricted version in which we also restrict it with the logarithm of the distribution in the whole database. So the first question I think that came into mind is, why do we need 9 anonymization techniques? Well, I think that this diagram allows us to understand it better. If we are interested in preventing re-identification attacks, then we can use canonymity. If we want to focus on preventing homogeneity attacks, then alpha canonymity can be the proper technique. And if we focus on the diversity of the sensitive attribute, then we have, as we have already seen, L diversity. Concerning L diversity, if we want a stricter measure for the diversity, we can use entropy L diversity or recursive CL diversity. And if we want to focus on the frequency and the distribution of the values of the sensitive attribute in the whole database and in a specific equivalence class, we have the notion of T-closeness, of delta disclosure privacy in the case that we have a multiplicative definition for measuring the difference between the distribution. And in case that we want to consider gain of positive and negative information, we have the definitions of basic beta likeness and even more robust enhanced beta likeness. So these 9 techniques are the one that we have implemented in the 2 Python libraries that we are going to review today. The first one, HANA, is a Python library that allows us to anonymize sensitive tabular data. So these 9 anonymization techniques are implemented, and as you can see in this screenshot of the code, we only need to define the hierarchies, the number of records that we allow to suppress in order to achieve the anonymization level required. In this case, it's a percentage of the records that we allow to suppress. And we can apply different functions. For example, here, LFAC anonymity for anonymizing our data, given the identifiers, the quasi-identifiers, the sensitive attribute, the value of K for anonymity, and the value of alpha. So the objective is to anonymize the tabular data using these 9 anonymization techniques, and we have further functionalities. The users can define and create their own hierarchies. We can work with multiple sensitive attributes. We can obtain the transformation applied in order to be able to generalize such transformation to a new data set with similar properties, or we can directly apply a given transformation, which means that we may be interested in saying, okay, for this data set, I want to apply the level 1 of hierarchy for this column, the level 2 for this other column, and so on and so forth. And this Python library, of course, can be simply installed, saying pip install HANA, and we can use it locally. But what we want to showcase today is how we can use this tool from the EOSC SIESTA service catalog. So we can go to the EOSC SIESTA dashboard, and here in the service catalog, we have our anonymization catalog where we have HANA, we have PyCanon, and TrustGoDP that I'm going to briefly mention at the end of my talk. So if we go to this HANA service, we can launch it, and we will be able to access to this interactive interface for performing the anonymization. Let me open this other service because while the container is starting, it takes a little bit, just some minutes, but just in case, this is the web application that we will be able to see, and we can upload the data that we want to anonymize. We can even retrieve the data that we have in our S3 storage in the platform, but let's start with a quite simple example, which is the adult data set. So we can upload the data, we can preview the data here, the age, work, class, education, different features that are available in this widely known data set, which is an extraction of the census. So of course here, our sensitive attribute will be the salary class. So let's go with the anonymization process. We can select the set of identifiers. In this case, we are not selecting any identifier because, as you can see here, all the features that we have can be insensitive, can be sensitive, or quasi-identifiers. So concerning quasi-identifiers, following the example that I showed you in the code, we can consider as identifiers the age, the work class, then also the marital status, the occupation of the individual, and finally the gender and the native country. We'll have then 6 quasi-identifiers, and the sensitive attribute, as already mentioned, will be the salary class. So then we can select the anonymization technique that we want to apply to this data set. We have here the 9 anonymization techniques that I mentioned. If we go to LFAC anonymity, we will be able to customize the value of K, the value of alpha, same for L diversity, and so on and so forth. So let's go with the simplest one, and let's apply K anonymity for K equal to 2. And then we will need to upload a file with the hierarchies for the quasi-identifiers. For example, we'll need to upload the hierarchies that we are interested in applying for the age, that in this case will be different intervals of different length to be applied. For example, for generalizing the age, I don't know, but let's say 18 years, we will generalize to between 15 and 20 years. And we need to do the same for the other quasi-identifiers that we have. I have these predefined hierarchies for work class, which is a categorical feature, then for marital status, then we have the occupation, which again is a categorical feature, so we have predefined these hierarchies in this case. For the gender, the same one, or we can even select to simply remove the value if needed, which we call suppress the records. And finally, the country. So then we are ready. We have uploaded all the hierarchies that we have for our quasi-identifiers. And let's wait a few seconds in order to get the data anonymized. Okay, so in this case, we obtained the data anonymized for the age, the work class has been suppressed, the occupation, the native country has been suppressed. So with quite a strict level of anonymization. And if we go back to reprocess the data, let me go back, and upload again the same data set, just for taking this into account. Okay, we have, again, the initial data. You will be able to see that we still have the quasi-identifier sensitive attributes. Here we have anonymity, and here we have the hierarchy. So you have seen that we had a quite a strict level of anonymization because we selected a suppression level of 0%. We do not allow to remove any record. So let's see what happens when we say that we allow to suppress the 20% of the data, and let's process the data again. So in this case, you can see that now this data has a better say because the age has been generalized but not suppressed to intervals of length 5, and we have not suppressed any column of the dataset. We have probably suppressed more records because in the previous case, we do not allow to suppress any records, but in this case, probably the data are more useful. So we can download this anonymized data, or we can store it in the S3 storage. So this concerns our first tool that we wanted to showcase today, and the second one is PyCanon. And PyCanon, concerning the differences with HANA, is a Python library that we can use to assess the values of the different parameters that we have seen for the 9 anonymization techniques. So we will be able to upload a data set and calculate the value of K for anonymity, of L for L diversity, and so on and so forth for the 9 anonymization techniques. We can simply install it with pip install PyCanon, and then it can help us to detect failures in the anonymization process. Again, we can deal with more than one sensitive attribute. We can calculate different utility metrics, and we can also obtain customized reports in JSON and in PDF formats, as you can see in this example here. We have for these quasi-identifiers, for this sensitive attribute, this is the report that we can obtain. And again, this service works in the same vein as HANA, so we can go to the service catalog and launch this service. I have already launched it here. So again, we can upload the data that we want to check the level of anonymity. So in this case, I'm going to upload this version of the adult anonymized dataset that we just generated with HANA. And then we need to select the identifiers, sensitive attributes, and quasi-identifiers. So our quasi-identifiers, the age, the work class, the marital status, the occupation, the gender, and the native country. And finally, the sensitive attribute that was the salary class. And here we can select the techniques that we want to check. We can select to check only the value of K for anonymity, checking L for L diversity, or we can select all the techniques available. We can use this apply generalization in case that we are dealing with more than one sensitive attributes, and then we can generate this report concerning the anonymization techniques that are fulfilled for this data set. And here we have this complete report, quasi-identifiers, and sensitive attribute, the value of K for anonymity as expected 2, because it's the one that we apply in HANA. And then we have the other parameters that are verified concerning the other techniques. And we can even download this report in JSON format in order to have it stored and to be able to compare different versions of the data and the anonymization level. So let me go back to the slides. So these are the main two tools that we wanted to present regarding data anonymization. Again, you can use the tools directly with pip install in your local computers. You can use it via the SIESTA dashboard in order to get advantage of the web application and also of the S3 storage. But also you can try to deploy this web application that we created in your own devices in order to use the web application without needing to upload directly the data. And finally, I want to briefly comment, I say bonus because this was not planned, on a new tool that we integrated in the SIESTA catalog, which is TrustGoDP. That is a tool that allows us to generate synthetic data using using differential privacy mechanism. So first, we need to take into account that differential privacy is usually applied to calculate aggregated statistics, which we call global DP. So we do not usually apply noise directly to the data, but we apply it, for example, when we are calculating statistics, we are getting histograms concerning our data, when we are reviewing the distributions. And it is not widely implemented for microdata publication because we will need to add a huge level of noise in order to be able to prevent disclosure of the data, and thus we will be damaging the utility of such data. However, we thought about using these differential privacy mechanisms, classic differential privacy mechanisms, such as randomized response, for example, for generating synthetic data based on the original ones, but adding noise with such mechanisms. So, of course, it's essential to strike a balance between the level of privacy that we want to achieve and the amount of noise that we need to maintain in order so that we have data that is significant. And we observed a lack of open tools for this purpose, specifically written in Python. So thus we implemented TrustGoDP that again can be installed via pip install and so on and so forth, and implements different differential privacy mechanisms, but to be applied directly to the raw dataset in order to generate a synthetic version. So of course we need a privacy budget, which is this epsilon here, that are greater than in the cases of global DP, but we are taking advantage of different classic differential privacy mechanisms such as Laplace and Gaussian for numerical records, or exponential and randomized response for categorical ones. Again, the functionalities are quite similar that for HANA and for PyCanon. We can simply select the column to which we want to add noise, the mechanism that we want to apply, and we will be able to create a new column with the values, let's say, with noise. And the interactive interface works in the same way. We can upload the data, then we can select the column that we want to perturb, for example, in this case, the age, which is numerical, the mechanism that we want to use, and then we will get here, this is the real value for the age, and the one when we add noise that you can find here in DP8. Again, we can download the data, and so on and so forth. So this is a new tool that, of course, is work in progress still, but already available for testing, both doing pip install TrustGoDP and also from the SIESTA dashboard. So in order to conclude, both HANA, Python, and TrustGoDP are already available using their web applications from the EOSC SIESTA catalog. HANA allows us to anonymize the tabular data, and PyCanon to check the privacy level based on such anonymization techniques. And work to be done is to include the privacy utility metrics in the web application, and also disclosure risk metrics that are available in the library, but not yet in the web application. So users will be able to check the privacy level, but also the utility of the data. And finally, TrustGoDP uses these differential privacy mechanisms for perturbing the data, but is suited for synthetic data generation. And of course, this is under development, and the first version of the web application is available. And finally, again, a bonus concerning not only directly data privacy, as we have been reviewing, but privacy in machine and deep learning. In the EOSC SIESTA catalog, you will be also able to find an implementation of a federated learning client. So we are also focusing on being able to provide privacy-preserving machine learning via distributed machine learning via, again, federated learning. So we can launch a federated learning client in EOSC SIESTA in order to perform a federated learning training using also the i4EOS platform. But that's another story, and we will be talking about this probably soon. And of course, you have several tutorials on the YouTube channel of the project. And that's all from my side. I'm now more than open to your question. So thank you very much. I don't know, Álvaro, if you want to show the slide or just in case. I don't know, it's my fault, but I cannot hear you.
Yes, no, sorry, I had the microphone muted. So yes, this is the Slido that you can use to ask questions. However, there are already some in the system. So let's go straight ahead. So there is one question about if it would improve the adoption of the tools to provide a graphical user interface to be used as a local application rather than running it on the cloud. What are the plans for this?
Yeah, the web application that I saw that is available in the portal, in the service catalog, it can also be launched in local devices using Docker Compose in order to have the interface directly and use it in our local resources, as I mentioned, just in case that one user does not want to upload the data directly to the platform. It can be used locally, even via the Python library, or using this web application, which we think that is more intuitive, specifically for users that are not quite used to use Python.
Okay, then we have another question about what is the difference between HANA and ARX?
Well, we started using ARX, but as it is written in Java and we found it very, very useful, we wanted to create this Python library because we want to have a complete pipeline to integrate it in a, let's say, data science workflow in which you can start with processing the data, getting the data, processing it, then you can apply directly these anonymization techniques using a complete Python pipeline with all the steps. So I think that most of the functionalities regarding anonymization tools that are implemented in these two libraries are mostly the same. Well, there are some differences, but they are quite similar. But the idea of doing this in Python is simply to be able to have a complete Python workflow.
Okay, and then maybe we can have the last question, that is about the hierarchies. How can one create the hierarchies for generalizing the data?
Well, I think that it depends on the kind of the type of data. For example, for numerical filters, for numerical columns, we have one function in HANA which is called generate intervals. If I'm not mistaken, it's generate intervals. It's available in the utils package in HANA. So you can say, okay, for this column, I want to generate intervals of length, whatever, with a minimum value of the minimum of the column and the maximum of the column. And you can generate the first level of interval, the first level of hierarchy for generalizing as an interval of length, let's say 5 for the age, the second level of length 10, and so on and so forth. For numerical attributes, it is supported, but for categorical ones, well, there are resources online for such hierarchies, but of course it depends in the case. For example, zip codes, you can remove the last digit of the zip code, the last 2 digits, and so on and so forth.
Okay, thank you, Judith. I think we can stop here, and if there are any outstanding questions, then we can catch up after all the sessions have ended. So
Thank you
now
very much.
thank you, Judith. Now let's move to our next speaker, which is Cyril Pernet. Cyril will talk about assessing metadata privacy in neuroimaging. Cyril is a cognitive neuropsychologist that has turned into a neuroinformatician. He is associate professor in translational neuroimaging, and his research bridges the gap between technology and experimental and clinical neuroimaging. Cyril has a long-standing interest in data sharing and reproducibility, as he could develop, for example, the section of the brain image data structure and was a steering group member. He's currently leading the Open NeuroPet project, an international group developing standards and open tools for pet imaging. And in this context, he has developed the public neuro.eu, a data sharing platform from brain imaging data in Europe. Since brain imaging data are usually considered personal data, he is interested in techniques and the tools for assessing and anonymizing such data. And today, he will present an analysis of neuroimaging datasets using a dedicated toolbox that is called MetaPribit, that, as I said, will be used to assess metadata privacy in neuroimaging. So, Cyril, the floor is yours. I can confirm that we can see your screen.
Great, thank you, Alvaro. Okay, welcome everybody. Thanks for attending. So metadata privacy in neuroimaging. I will quickly talk about brain imaging. And the way we do brain imaging is using structured data sets. So the advantage in our field is that there is a standard. So we know in advance how things are named and organized. So I took an example here from another repository, which is called Open Neuro, and that's in the US. And as you can see on the screen, basically the organization is always the same. We have subjects and then different folders and different names, and all of these are very standard, which is very useful. And Robert will probably talk a little bit about these too. I said the data are structured, and this is the same type of data which are collected for clinical use, even though this is for research, the context in which we are working. This is the same kind of images. They are fancier, more complex, curated, whatever. And that's because this is research, but fundamentally the same. As an example, for instance, one of these files would look like this, which is an MRI. So that's the brain of one subject. And here, this MRI, as you can notice a little bit at the front, for instance, has been cropped. It's all black. So that would be this part of the part. And then you can see here, it's like kind of distorted. And that allows to remove the face. And that's simply because the face can be reconstructed digitally. And then you can match it with databases. So even so, the brain pattern is unique, a bit like your fingerprint. And it means also, for instance, if you have patients with specific lesions, they may have unique characteristics. And then a background attack, for instance, would allow to recognize those people. So in general, in Europe, we can still see these data as sensitive personal data. So no matter what we do, no matter what kind of techniques we are applying, typically, this is considered sensitive personal data. And this is why we have tools like the bit scramblers that Robert will present after me, which allows to develop analysis pipeline in a way which is completely anonymous. But in this case, what everyone is concerned about these images, and for good reason, I focus on associated data. Because when you analyze brain imaging data or medical data in general, you cannot do anything without the metadata that goes with it, about the images, but also about your participants. And so for me, is this tiny file there. And that basically makes the link between what Judith is presenting about tabular data and what Robert is going to do next. This is also tabular data. So if I click on this file, it will look like this. And so you can see this is tabular. We have a participant ID, age, sex, and then we start to get into all sorts of other interesting parameters and variables about these particular subjects. Some of it being demographic, some of it being clinical data. So what's important in this file is the participant information. The participant typically would be a TSV file, and also it has this little sidecar which allows to describe what each column means. And we will always have like age and sex, and typically also handness and education level. And in many cases, these also include sensitive personal related data related to physical attributes and mental health. And these, in a way, are essential characteristics because when we analyze this type of medical data, well, we need these because these are the characteristics that we want to study, how this influences your brain structure, what are the specific effects of different variables on brain so that we can act on brain via stimulations or via drugs. Therefore, we must have this type of information, which creates a tension between the privacy. So we don't want to put too many of those, or we want to reduce their information content. But in that case, we would reduce utility. So this is this kind of trade-off that we need to have between privacy and utility. So how did we assess tabular data? So we created that meta-previds, which is mostly a wrapper. So we use the same basic metric, K anonymity and L diversity that Judith is using. And then because if there is something very specific in these kind of data sets that I will come back to, which is the fact that all these informations are related. So when you have a patient which has, for instance, a low performance in language in a given test, you might also have a low performance in other associated tasks, and that creates profiles, right? And so now if we are talking about a multivariate vector, when we look at one particular row, not columns, then we have a row, and that allows to identify uniquely people. And for this reason, we used a different type of metrics. So we developed one called K global, and also something which is called sample unique detection algorithm, and also privacy information factors. One is used by the UK government at the moment, and the last one is something developed by Australia. What meta-previds does is wrap those 4 metrics, plus we add ours that we developed, and then we leverage also this JSON file, because these JSON files are little helpers. Typically, we describe what type of variable information the information contains, and so it's easier to do these kind of separations that Judith was talking about, also between categorical, continuous variable, if they are sensitive or not. Of course, the wrapper, we can also have a GUI, but typically you can also just pip install it, and then as is shown on the screen, in a couple of lines, you can get all of your metrics. And so the goal of this was mostly to write a single wrapper in one place, again in Python, because then it's open and it's easier for everybody to use. And if people want, they can also have a graphical user interface. A key aspect, again, is these repeated measurements, because everything is correlated when we look at one patient. And so sure, K and L works, but they work at the variable level. And then this pseudo algorithm is interesting because what it does is find these unique combinations of variables. So it takes every variable and then associate them one by one. So you start by pairs and then triplets, quadruplets, and so on. And that gives a score, but per subject rather than per variable. And the same thing with PIF, which basically is something that looks at how much information is gained by adding one variable to an entire set. So you take one variable off and you say, well, what's my posterior probability once I add this new variable? So it's information-based. And this K global that we also created was there because we realized it was, even though those informations were useful, it was difficult to identify the interrelationship between all these correlated variables. So there's plenty of metrics. This is not the only metrics available, but most of these metrics, they do something about the multivariate pattern, but they don't help people to figure out which variable in this multivariate pattern has a higher weight. And so that's what we wanted to quantify, is how much a variable weights relative to every other variables in the anonymity balance. So it's still K, like what Judith presented, but in a different context. Just to give you an example, from this exact data set I was showing a couple of minutes ago, we have this KI global that we're computing. It's basically the difference between the number of unique combinations of variable in a data set versus the unique number, but not that variable of interest. So then we have a relative difference. And then we simply normalize it by the number of unique value for this particular variable that we want to look at. So if we go back to this table that I showed you earlier, here I selected age, sex, handness, educations. There is also the background, socioeconomic background of people. And then in this data set, they also look at sexual attributes, sexual attraction, and also gender identity. And so we can get a given K global value for all of these attributes. So we get, again, the local K per variable, and then the global K, the contribution to the entire set. And so if we start by, for instance, looking at age, we're like, okay, we need to change age because it has a very low K value. But what impacts that would have against everything else? So for instance, if we focus on age, we want to increase K anonymity. So it's simple because in their example, they use age with, like, instead of rounding values, they use down to the month, which of course means everybody is unique. So we simply take age in years. That makes things a little bit better. We get to 2. So we reduce the unique value. And so there is a move of also increasing basically the minimum group size. But what it does is that because it's less unique on its own, it has a better K, but it becomes more important for distinguishing participants when combined with other variables. So the K global increases. It means that age on its own is better, but on the other hand, it makes classes more correlated to each other. And another variable of interest, which in this case works probably a little bit better than just playing with age, would be to change the socioeconomic status. Here we simply change from 9 beans to 8 beans. So again, we reduce the granularity. And by doing so, the K anonymity went from 23 to 13, which means it's still okay. We reduce a little, but that works. But as for age, it has a huge impact on uniqueness. So we can see that the total uniqueness, for instance, has a reduction of 36%. So we move from 700 to 400. But what's really interesting, and that's the whole point of this type of metrics, is to try to understand the relationship between variables. Because again, when we look about how humans behave and we look at their traits, then everything is correlated with everything. So we want to be able to look at this impact. And here, for instance, when we look at the socioeconomic status, it also impacts education. And it makes sense because typically, if you come from a background with a lower socioeconomic status, you also have a lower education, and the other way around, right? And so we cannot just keep considering each variable independently. It's important to be able to assess how, if I change one, it will impact the others to create a data set overall, which is well anonymized. And that's this interdependence between variables that is important. So now that I presented this, I wanted to show what we obtained. So from this database, what we did is just to say that we actually went to get that published last month. We look at 8 datasets, and across these 8 datasets, we actually could re-identify 2 people, which were relatively easy to cross-link because they are mostly in the US. and we have like a census where you can figure a lot of things out. So it's still a very low percentage, but still we could identify this, which means that once we could see that these people were at risk, the whole record were updated. And then in the paper that I just mentioned, we only actually published on 6 datasets, just to make sure that nobody tries to go back to the data they might have downloaded and figure out what difference is in there. And on these 6 datasets, what we see is that interestingly, clinical scores, which is typically what the lawyers have been very worried about, pose minimal re-identification risk. And that makes sense because the linkage is almost impossible because there's no database related to something about your IQ or something about your performance in a memory test. And also there are continuous values. So typically it would be nicely distributed. You don't have visible attributes. And again, similar to the kind of things that Judith was talking about, demographic variables were really what was at play here: age, sex, assigned birth, sexual orientation, race, income, and geolocation. That are basically all the things that every time in each dataset we could find some problems with. So what are the lessons learned is that privacy tools are necessary but not sufficient. So we can deploy these tools. And I think the things that Judith is presenting is really, really nice. A lot of different options and we can increase those. But so they are necessary because they allow to identify variables in an entire dataset. So the dataset I just talked about today, for instance, is something like 900 people, something 50, 60 variables. Of course, it's very difficult to navigate those tables as a human. So we want tools to help us to flag the interdependencies between variables and flagging outlier participants, which is something that SUDA and PIV do very well. But it's not sufficient because identifying sensitive variables and applying privacy change, such as rounding values, aggregating different classes, or adding noise, if it's a continuous variable, this requires understanding of the variables to keep the utility, meaning that humans, at least humans for now, with domain expertise is required to do the anonymization. We can provide tools and we have developed tools, but they need to be operated by people with domain expertise. Thank you for your attention.
Thank you, Cyril, for your presentation. I remember, let me share again the screen with the Slido. Okay, so this is the page for the questions. We have one question that is, what is the difference between the tool you just presented and HANA?
The two metrics K and L are exactly the same. I mean, we work with Judith as well. But the K global at the moment, the K global is not in Anjana, so that's something that we tested specifically for neuroimaging because we know how correlated that was data. But it'd be, very easy to incorporate it. SUDA and PIV are two very different methods that we tested because of the specificity of this interrelation between variables. So I don't know. I haven't checked in a little while how Anjana is dealing with the correlated variables, but those extra metrics that you can get it from our tools, but they're also available from other libraries. You can just get them from GitHub and pip install, and they are very specific to these correlated structures between variables. And that's really what these extra metrics are doing.
Okay, thank you, Cyril. And we have another question, and it's about if this tool can only be used for neuroimaging data, because there are some functions that are interesting for other kinds of data, of data sets.
No. What it does is it takes any tabular data. So, it does. It's just a table. What it does, what's specific to neuroimaging, is because the tabular data always come with a dictionary as a JSON, which is not always the case in all fields. And so it leverages. If you don't have the JSON, it still works. But if you have the JSON, it makes things easier because it can categorize and the different variables automatically for you, because the JSON explains typically so little, right? Sidecar dictionary that would say, oh, this is categorical variable, this is continuous variable, this is what it means. It also helps humans to understand what are each variables, each columns, what they mean.
Thank you, Cyril. So then let's move to our next session. Thank you again, Cyril, for your talk. The next talk will be given by Robert Oostenbel, a neuroimaging researcher at the Donders Institute for Brain, Cognition and Behavior at the Radboud University Nijmegen in the Netherlands. He is the leader of the medical imaging use case within the SIESTA project, and his research focuses on data analysis methods for EEG and MEG with applications in cognitive neuroimaging. In this context, he's the author of Field Trip, one of the most widely used open source EEG and MEG analysis toolboxes. He designed the Donders repository for data sharing that now has become the Radboud data repository, and he served on the steering group for the brain imaging data structure. He has multiple publications dealing with sensitive neuroimaging data, and today he will present how scrambled or synthetic data can contribute to better reuse of sensitive neuroimaging datasets, talking about one tool that we have developed within the project that is called Bit Scramble. So Robert, the floor is yours. We can see your screen. So thank you, Robert. Let's go ahead.
Okay, thank you, Alvaro. Welcome to everyone. Nice of you attending. So I'm going to explain how we use Bit Scramble to generate pseudo-random or synthetic data sets. So this starts from the medical imaging use case in the SIESTA project. And considering medical data in general, there are way too many standards to mention. If we zoom in a little bit, then we can see that medical databases often relate to long-term patient records, interviews, and reports, and they link to data elsewhere. But if we look more at the specific devices that are used, then it becomes a little bit more tangible. So for MRI and other imaging data, we have DICOM standards. We use PAC servers. For microscopy, we would use vendor-specific format, but also standards such as OMATIF. Again, EEG data, like scalp recordings of potentials of brain activity, we have many vendor-specific file formats and databases, and the same for genetics. These medical records, they comprise a complex and inconsistent longitudinal database that's spread over many systems in hospital and in clinical settings. But if we take them out of their clinical environment and if we use them as research data for a study, then we're taking a subset of all this medical data that we wrangle into a better organization with an eye on the analysis tools that we will subsequently be using on the data that we have. Zooming in on a particular type of data, neuroimaging data or imaging data from the brain, there are still too many formats to mention. So each imaging modality, each device and each vendor has its own data formats. But if you look at neuroimaging data standards, then it becomes more tangible, then it becomes more managed. There's not too many of them. So DICOM is one of these standards that is mainly used in clinical settings. DICOM files consist of 2D slices with imaging data and a lot of often repeating header information. For neuroimaging data analysis tools, DICOM is not so often used, but rather data is converted into the NIfTY format, which is mainly used in research settings, where the NIfTY files contain 3D volumetric data or 4D data, including a time axis so that we can track how brain activity changes over time. And these NIfTY files are relatively compact, they have smaller headers. As Cyril already mentioned, since around 2016, we have the brain imaging data structure, which is a standard for neuroimaging and specifically for cognitive neuroimaging. It's like mainly developed for cognitive neuroimaging, but can also be used for clinical neuroimaging. Initially, it was developed for MRI data, but now it has been extended to also cover positron emission tomography, microscopy, EEG, MEG, functional NIRS, etc. What BITS does is it complements the brain data by metadata in tab-separated value files, TSV files, and JSON files. And BITS focuses on the interoperability and the reusability of data sets in FAIR. So BITS doesn't solve everything, but there's like other things that solve other aspects. For example, OpenNeuro, a repository, which is built on BITS datasets, which then adds the findability and the accessibility. When recorded in experimental medical research and cognitive neuroimaging, then the experimental design and other metadata are crucial for data reuse. So it's not only about the images, but it's knowing about in which situation those images were recorded, what the participants were doing, what the clinical state of those participants was, etc. So further zooming in neuroimaging data in cognitive research, then we have the imaging, which is complemented with other types of data. And that other type of data, that is, for example, tabular data, including demographics, age, handedness, those type of things, scores on general clinical or psychological tests, such as the IQ, and performance on study-specific tasks that relate to the cognitive function that is being investigated. And furthermore, neuroimaging data can also be complemented with links to non-tabular data. So the tabular data is what we've been looking at in the previous two presentations, but the non-tabular data is, for example, the physiological signals from scalp-recorded EEG or MEG, anatomical functional MRI imaging data, but also the behavioral data, the responses of the participants to certain tasks, movements, eye tracking, and there's a lot of experimental design and description of the intervention in the non-tabular data as well. The medical imaging data itself can be processed and diagnosed, and thereby it can become a tabular result, but it always starts as a non-tabular data. So the challenge that we have in imaging research is like translating this non-tabular data into something that is like more easily interpretable. So I'm focusing on the part where the medical imaging data is not yet a diagnostic, or the outcome of a diagnosis, but it's really the images. So an example of this is, for example, this data set where in the background you see a tabular data where a number of participants are shown. Each participant has multiple variables. In this case, it's a patient versus control study. The patient, this is a Parkinson data set. Age has been specified, sex, handedness, etc. have been specified. But each of the records in this table, each of the rows in this table is linked to anatomical imaging data and is linked to physiological data that was recorded, for example, with scalp EEG, where also in the physiological data, the participant was doing a specific task, for example, a cognitive task in which the participant had to press buttons, and these buttons could be either wrong or correct, and then resulting to like a reward or to a penalty. The challenge that we have with medical or neuroimaging data is that it's the identifiability. The measurements result in very detailed data that could be considered as a fingerprint. And as Cyril already showed, is that the facial details in an MRI scan are actually quite easy to extract, and which is why for an MRI scan we often use defacing strategies to not share the facial details. But that in itself is not sufficient, because the folding pattern of the cortex of the brain can also be considered a fingerprint. It's unique, it's stable over time, and it could be used to link to external databases. And as Judith already showed, it's also the combination of demographic data can be used to link data to external databases. So when the data is pseudonymized, so in the absence of direct identifiers such as the name or address, it is non-trivial to decide whether data is personal or anonymous in GDPR terms. Tools such as Anyana and PyCanon and MetaPrivBits can be used for doing this, but I'm going to describe a different approach that we use in the analysis of neuroimaging data. If we consider like whether data is identifiable or not, then there's a few characteristics to consider. Like, are the potentially identifying features like easily and unambiguously extracted from the imaging data, such as the face, which is easy to extract, cortical folding pattern already more difficult. Is it constant over time, and can it be mapped to an external database like fingerprints or like faces, photos of faces that you would, for example, find on Facebook? The challenge that we then end up with is very similar to Guess Who. We have multiple elements on which we can identify a person. For example, a red-haired person that is like partially bald and male. We could basically single out such a person. Some types of data by themselves are not identifiable, and when properly pseudonymized and de-identified, and when we're not disclosing the key, the data becomes anonymous. But the more metadata is available, the larger the chance of the data being identifiable. Again, going to this Parkinson dataset that I just showed, like if we'd say the dataset includes a 73-year-old woman with Parkinson's disease, left-handed, who participated in a neuroimaging study at the Donners, which is implicit knowledge by the participant being part of this dataset. That also then discloses like that the participant was likely to be of higher socioeconomical status, which happens to be the bias in the sample that we're collecting from. Like we are better able to reach those participants, and probably it's someone who's living in or close to Nijmegen. So that means that even though we're only disclosing 4 items explicitly, there's also some implicit items that are being disclosed, and such this example provides 6 items of information with which the risk of this participant being identified increases. So this poses a problem for the reuse of medical or neuroimaging data. What we considered in the medical imaging use case in SIESTA is the situation where we have medical data or medical neuroimaging data that is being acquired at one institute, and where we have researchers that want to use that data which are at another institute. So the ones that acquire the data and are responsible for the ethics and the legal aspects are the data rights holders, and the researchers that want to reuse the data are the data users. If the data is used in the same setting as where it's acquired, it is usually not such a challenge because the ethical provisions and the legal provisions are already in place. But if the data rights holders and the data users are separate from each other, then we have a challenging situation. And in that case, we need contracts when data is shared by the data rights holders to the data users. So those contracts can be called data transfer agreements or data use agreements or licenses. But what we want to focus on is the situation where the legal hurdles of making a data transfer agreement or a data use agreement is not taken, and also where the technical hurdles or the scientific hurdles of anonymizing data sets are also not being taken. So what we want to focus on is the reuse of neuroimaging data without actual sharing. And that's where federated analysis comes in play. The idea behind federated analysis is that the data rights holder performs the analysis on behalf of the data user. And it's only the results of the analysis are shared from the data rights holder to the data user. So if we look at that in a more schematic fashion, then here on the left you see the sensitive data that is the responsibility of the data rights holder. And then we have the data user. The data user knows about the data set because sufficient details of the data set are apparently known. And the data user has a certain analysis in mind, and that analysis is then implemented in an analysis pipeline, which would result in group level results. Of course, the data user would preferably have access to the sensitive data, but that is not possible, so there's a hard boundary between the two. What we do in federated analysis is that the analysis pipeline is implemented by the data user, is then subsequently handed over to the data rights holder, and the data rights holder executes the analysis pipeline on the sensitive data on their own premise and only shares the non-sensitive group level results. So this is if we look at this, the challenge is like, how do we implement this analysis pipeline? I'm assuming here that it's a new research question, so there's no standardized analysis pipeline that simply can be executed. So it's the data user and the data rights holder have to work together in order to make this possible. So how to implement the data analysis pipeline? So as I already said, we have a complex format of neuroimaging data and metadata, but this is where standards come into play. So we can have details, we can share details of the data without sharing the actual data. So very simple would, for example, be just to share documentation, like in a data descriptive paper, a README file, PDF documents, etc. More specific, we can share a data dictionary that explains the columns and the values within those columns. So for example, in this case, we have the participant ID, we have the group, which is a clinical categorization with 2 values. We have age, which is specified in 5-year spins, which is like the sex assigned at birth. We have the levodopa equivalent daily dosage in milligram per day, etc. So the description of the columns, but also the description of the values that can be used in those columns is the data dictionary. And that is what Cyril already referred to as a JSON file that accompanies tabular data in a bits data set. So the neuroimaging data itself is not tabular. That also means that a simple data dictionary cannot really apply to that type of data. And also tabular format is not sufficient to explain the complex relation between the data and the experimental design. The brain imaging data structure specifies how data is organized, and it also specifies how metadata can be added as a series of TSV and JSON files. And the presence or absence of certain data in one participant or patient, like for example, a certain scan, whether that has been done or whether that has not been done on a particular patient, that would be indicated by the corresponding file simply being present or being absent. So it's not tabular information by itself, but it's the whole construct of all the files that conveys information. The implication of that is that the data user, so the researcher that is implementing the analysis, needs to see the whole file and directory structure, plus all the metadata to fully understand the data set and to be able to implement an analysis pipeline in the domain-specific tools, such as FSL or Fieldtrip or SPM or M&E Python or any of those tools that we use for neuroimaging data. Here I'm showing a small like snippet of a bits dataset. Cyril already showed them previously in the Open Neuro platform environment. For another example, so here is a dataset where I'm showing like 3 participants, like 3 subjects, where for subject 1 we have anatomical and functional scans. So that's the second level of the directory structure with anatomical and functional. And we have the data files themselves, and we have metadata. The important thing here is that in bits, it is prescribed that data files are accompanied with a JSON file, which explains the corresponding data file. So this file explains this one, and this file explains this one. Furthermore, we have, in this case, we have a functional data set where the task was apparently to finger tap, and the like functional MRI data was acquired. Again, we have a JSON file that explains the settings of the scanner during which the finger tapping task was performed and the bolt data was recorded. But we also have an events file that explains the task, and we have an events.json file which explains the details of the task. For example, details on what the instructions were, but also what the fingers were that were used in the tapping, the exact timing of the tapping, etc. So it's this constellation of data that is necessary for being able to implement a certain analysis pipeline. So on the one hand, we have the sensitive data that's the responsibility of the data rights holder. On the other hand, we have the data user that wants to implement a pipeline and therefore needs to have access to details of the data. And this is where we have implemented BitScramble. BitScramble is a Python-based tool that takes sensitive data or potentially sensitive data in the bits format, and that creates a pseudo version of that same data set, which then can be revealed, can be shared, because it actually does not contain the real data anymore, and that can be shared with the data user, such that the data user can implement the analysis pipeline and get evaluate whether results can be computed, even though those results are not the real results. So these are also pseudo results. So you can imagine that correlations in the data are going to be totally destroyed. So the correlation that you get as a result is going to be 0, but at least you can go through all the steps of the analysis pipeline to implement the code, the scripts that are necessary for the analysis. So what bit scramble does is it takes the original potentially sensitive personal data in bit format, and it creates a scrambled anonymous data representation out of this, where all the files and all the like the whole directory structure remains intact. So the scrambled anonymous data can be shared with the data user, and that means that with this pseudo data, the data user can implement the analysis pipeline, and once this analysis pipeline has been implemented, it can be handed over to the data rights holder, and the data rights holder then can apply the analysis pipeline to the real sensitive data, can review the results, and then can subsequently share those results with the data user, because at that moment, it's the data rights holder that can assess that there's no non-sensitive group level results being outputted by the analysis pipeline. Bit scramble is implemented as a Python package. It runs in the command line, and it can be installed using simply using pip install, which installs it from PyPy. This works on both Windows, Linux, and Mac OS, and it comes with online documentation. It comes with command line documentation as well. The scrambling depends on the file type. So as you can imagine, the type of scrambling that would be necessary for one characteristic of the participants, like for example, the sex or the gender, would be different than the scrambling that would be required for imaging data. So what we have implemented so far, and these are definitely things that we can further extend, is that for tabular data, we scramble the values over the rows, so within a column. So that means that the age, for example, the age column, all the values of the age are shuffled within that column, thereby destroying the relationship within one participant, which is one row of the data. We also have we can have data that's represented in JSON files, so where these values can be replaced by others, where certain key value pairs are removed if they're potentially identifying. And more importantly, for the imaging data, for example, for the MRI, we can add noise, or we can even replace the whole image with white noise, like with spatially distributed white noise. Or we can apply different range of spatial deformations to the images, where the spatially deformed images are not identifiable anymore, but still do allow for the standard analysis tools such as FSL and SPM to be applied to those images. Furthermore, we have physiological data such as the EEG, where we replace the time series for each of the channels with a random time series, where we again shuffle all the samples such that the temporal structures remain intact, but it's the details and it's the spatial correlation between different channels that loses all the information, still allowing for analysis tools such as FieldTrip and MME-Python to run on the analysis, but probably resulting in power spectra that don't really have the peaks at the physiological frequencies where you would expect them. Furthermore, the directory structure of the scrambled data remains the same as it was initially, and all the metadata is still there, and also all the files are still there, and they have a content that in principle allows for executing analysis using the domain-specific tools. All the correlations between the variables will be lost, and numerical results of the analysis are not meaningful. The implemented pipeline, so the pipeline that has been implemented by the data user, can subsequently be shared and sent to the data rights holder, who then applies it to the real data. So this is not yet all, because there's also the step of, like, if the scrambling really ensures the privacy in the scrambled data. The idea is that the pseudo data is anonymous. Actually, it can be like nonsense data. But for that, we have also implemented a tool which is called .leak, which can be used to analyze the information leakage between the two data sets in both the neuroimaging and in the tabular data. But I'm not going to talk about .leak today, but it is something that if you're going to use this tool, then I think you should see them as complementary in making the scrambled version and evaluating that the scrambled version can be shared as like non-anonymous pseudo data for pipeline development. So to summarize, sharing data is challenging. It requires legal agreements between data participants, data rights holders, and data users. And as we've seen in the previous two talks, sharing data can also be done after properly anonymizing data. But that in itself is also challenging because of the complex structures in the data, requiring quite some expertise in making anonymous data. Federated analysis, however, allows for reuse of data without actually sharing the sensitive data. And that's where these two tools come in. Bit scrambles allows for sharing enough details of the sensitive data set to allow the data user to implement the analysis on that data. And that leak allows the data rights holder to evaluate the information leakage and to ensure that the data is anonymous and therefore can be shared with the data user for pipeline development. The execution of the analysis pipeline and the evaluation of the results remains in the hands of the data rights holder. Thereby, we can ensure that there's no sensitive data being leaked to the data user or to the wider public through publications. That's what I wanted to present. So thank you for your attention.
Thank you so much, Robert. Again, let me ask you some questions in the Slido. So there is a first question about MRI scans. They ask is if from an MRI scan not anonymized, an attacker can identify a given individual.
Mm-hmm. Yeah, yes, that is correct. Like if, of course, it depends on the characteristics of the MRI scan, but an MRI scan of the head includes the face. The face is publicly quite easy to find in other accessible databases, such as Facebook or online with Google image search, you can find a lot of faces. So facial details are very easy to re-identify. Of course, if you have an MRI scan of another body part, like for example a knee or the stomach, that will be much more difficult to re-identify it because those body parts are typically not available in external databases. But it's in neuroimaging that we often have the face included. Defacing helps to reduce the chance of re-identifiability, but that does not necessarily guarantee that the data is anonymous. So it only makes the data less identifiable.
Thank you, Robert. We have a couple of more questions. One is, how does bit scramble ensure that the statistical distribution of the features is preserved?
So we have, for each of the data types, we have implemented a number of scrambling methods. Some of them preserving the statistical distribution of the data, whereas others not preserving this. So for example, if we replace the EEG data by random noise, we can make sure that the mean and the standard deviation of the random noise is the same as the mean and the standard deviation in the original EEG time series. But that does not mean that we have the same spectral structure, for example. Of course, we could, in this case, implement more sophisticated pseudo data generators that generate random time series with identical spectral structures, like for example, have the alpha peak, like the occipital alpha peak in there as well, whilst still not being identical to the data that was shared. So for the tabular data, we're retaining the internal structure of the data by simply shuffling, so not by replacing numbers. But it's for the imaging data and for the physiological data, we're actually replacing those numbers, or we're shuffling those numbers in such a way that the structure gets lost. We're also working on the paper to evaluate the consequences of that shuffling on the analysis tools that we subsequently apply to the data to make sure that the data is that the scrambled data is still of sufficient utility to be able to implement the pipelines.
Thank you, Robert. So then we have two more questions. One is to you, and another one I think it's more to me. So one is what is the difference with a bit pseudonym? That seems to be another bit anonymization tool.
So the bits ecosystem is a very large ecosystem. There's like a lot of tools are being developed for processing bits data sets. As Judith already explained with PyCanon, we're making, so like within Siesta, we have developed tools for making anonymous versions of tabular data. And as Cyril has explained, with metapriv bits. We've also made tools that go beyond that and make it specifically for bit data sets. So I would say the tool that was just mentioned, Bitsonimi, is more in the line of the metapriv bits tool that Cyril presented. So it is about making data anonymous such that it can be shared. Whereas BitScramble is actually for making data sets that are not that are anonymous, but actually that do not contain any personal data anymore, and actually that do not contain any useful data anymore. So it would be more similar to sharing data set where the data has been like totally blacked out, for example, like not only putting the bar over the eyes, but basically just replacing the photo with a black image or with a noisy image or a random image of a head. So as such, BitScramble takes a much more extreme approach for scrambling than Bitsonimi.
Okay, thank you, Robert. I think it's clear. And then we have one last question. I think this is a very pertinent one to close this first three presentation block. That is, if will all these tools be integrated in the EOS catalog? And the answer is yes. We are in this moment in the process of registering all of them through the EOSC Beyond Innovation Sandbox, in order to make them available either via standalone tools whenever this is possible, or via the whole Siesta platform registration in cases where it is not possible. So stay tuned, because this will be ready and integrated and available quite soon. So thank you again, Robert, for your talk. Let's move ahead with our next speaker, Fernando Aguilar. Fernando Aguilar is a scientist at the Institute of Physics of Cantabria from the Spanish National Research Council. He has experience with data science, advanced computing, and dealing with research infrastructures, being specialized in the design and operation of FAIR compliant data repositories and services. His work focuses on supporting the full research data lifecycle, from acquisition to integration of heterogeneous datasets. He has participated in several EU projects along these lines, such as Indigo Data Cloud or Extreme Data Cloud, contributing to repository integration, metadata standards, and also semantic interoperability. He's now coordinating the Goyas project that is developing a FAIR by design system for remote sensing, and he's the main developer of the FAIR EVA, which stands for Evaluator Validator Advisor, that will be presented today. Also in EOS SIESTA, he's participating in the analysis of the tension between the FAIR principles and sensitive data. And today, Fernando will talk about where should we publish our data and how should we publish our data, repository types, trustworthy evaluation, and FAIR metrics in practice. So, Fernando, if you can share your screen, the floor is yours.
Thank you, Alvaro. Yes, let me share my screen. Okay, I hope you can see it now.
Yes, we can.
Okay, so let's begin with my presentation. And the answer to where should I publish my data, the short answer is I don't know, but I hope I can help you providing some information and that helps you to decide. So we will talk about different repository types, how we can evaluate the trustworthiness of the digital repositories, and also we will talk about the FAIR principles and some metrics. So whenever researchers and data producers have some new data, well, we often ask ourselves, where can I publish this data set? Is the repository trustworthy enough to support my data? Especially for sensitive data or when they are anonymized, we can ask ourselves how open should be that data. And of course, in the context of the European Open Science Cloud, how fair is my dataset? So in this presentation, I hopefully provide more light on this sense. And it is especially relevant for a sensitive dataset after the different tools that we have seen, also anonymized dataset, and in some regulated domains. So we can categorize the repositories in different, different ways and in different types of repositories, but I think this is the most proper one. We have, for example, institutional repositories that are basically managed by universities or research institutions that are especially focused on the data, and not only data, but also research outputs from the researchers that belongs to that institution. For example, at the Spanish National Research Council, we have Digital CSIC, that is very focused on open science. We have also in different universities, like the University of León, we have Bulería, and in other research institutions like KIT, we have Radar for KIT. If we think about specific domains, there are a lot of different not only repositories, but also data portals or data spaces where we can find dedicated data, that is disciplinary data. We have, for example, the Global Biodiversity Information Facility that is oriented to host biodiversity data. We have at CERN the Open Data Portal where you can find the data from different experiments, or data coming from remote sensing. We have the Copernicus Data Space Ecosystem that provides not only data, but also different tools to manage and to process the data. And also at international and especially European level, we have general purpose repositories that are not belonging to or focused on a specific institution, but they are open to host data from different types and different disciplines. For example, of course, you probably know Zenodo, that is European, but it hosts data from at international level, and we have also Figshare. All those kind of repositories has their pros and their cons, but well, depending on the characteristics, you should choose one or another. I have to say also that in general they are not like a specific software, but they reuse some open source initiative like a DSpace or Dataverse in the case of general or institutional repository, and specific tools or software like Atlas of Living Australia for the case of the GBIF. It means that there are different repositories that are deploying the same software behind. So we have some examples here, but how can I find repositories, or how can I find data that is similar to that I want to share? Okay. On one hand, we have aggregators that basically connect with different repositories and provides you a search engine where you can just find some data in one single site. This are the case of Recolecta in Spain, that is the aggregator of the Science and Technology Foundation. And we have at European level OpenAir, that also connects you to different institutional, university, etc. repositories, and allows you to search in one endpoint different data. This is useful because you can try to find some data that is similar to yours, and then try to know if you can use that repository or a similar one. On the other hand, we have some registries like RE3Data, that is the Registry of Research Data Repositories, that are basically a catalog of repositories where you can find specific information about this list or types of repositories and try to know more about the features that they support. So whenever you go to the RE3Data portal, you can search and you can filter depending on the characteristics that you want to check, and you will see something like that. So you can try to find the type of content that you want to share and to limit the list of repositories that support that type of data. And you can also search among the list of more than, I think they are more than 3,000 repositories at global level. And you can, well, check exactly which kind of features it supports. For example, you can also filter to know if that repository have any kind of certification, like Core Trust Seal that I will talk about later. So this is one good way to decide which repository it's suitable to host my data. At EOSC level, which one of the goals is to develop and to implement a web of fair data. This ecosystem of repository is very important, but since they work separately, they should provide some interoperability feature to facilitate the reuse of the data. It should mint identifiers to the research outputs and use different metadata standards that could be compatible to be integrated. And also provide some machine accessibility that allows or facilitate the automatic processing of the data. Also in this EOSC context, one of the focus is to provide open data and open research outputs, but in some cases it's not possible. So some of the repositories should also support control access to allow the control of the people that have rights to access, and also restrict. We have some examples, as we have seen in the previous presentations, about sensitive data. In one of these disciplinary repositories or data portals where we collaborate with, this is a Global Biodiversity Information Facility, and it also includes sensitive data. In this case, for biodiversity, sensitive data typically refers to the occurrence of some species that have some risks depending on different things. For example, endangered or protected species. For example, for that is the location data is sensitive because you cannot share the precise nesting location of some protected birds or the breeding sites of some endangered amphibians. Also, species that are targeted by collectors that implies illegal trade, like for example orchids or rare orchids or some exotic pets, and also other ecological fragile habitats like coral reefs or rare plant species habitats. The solution that provide for that, in this case GBIF, is they recommend some best practices that includes the spatial generalization and also restricted coordinates, and in some cases control access. But in any case, they are transparent in this case because in the metadata they exactly details which kind of anonymization or generalization they are applying to a specific data set. So that information is available on the metadata. That is after that very useful. Regarding sensitive data and also in general anonymized or research outputs, we have different access models in these repositories. We have, for example, the open access, that is basically that the data is openly available without restrictions, so you can just go to the repository and get the data easily. We have embargo periods, which means that the data is temporarily restricted and become public after a specific period of time. This kind of access is used for publications whenever you are working on your results and you want to protect temporarily your data or you are working in your thesis and you want to have the data unpublished yet, then you can select an embargo period and it will be open access after the time that you have defined. We have also control access that, well, some committee or some mechanism needs to be applied to grant access after some request from the stakeholders. It is used, for example, in social sciences or clinical data sets. And we have also secure access where the data can only be accessed within secure environments and with strict controls. That it implies sensitive data or other kind of data. And the choice on selecting the access model depends on ethical constraints, but also the legal requirements or sensitivity of the data. So, to select which are the repositories that can support my data type and depending on the type of the data, we have the concept of trustworthy digital repository that, well, we have different definitions for that, but one of I like is a repository whose mission is to provide reliable long-term access to manage digital resources for its designated community. And what is important now and in the future. So trustworthy data repository means not only the access of the data, but also the preservation of the data. So in practice, this means that repository should demonstrate that it can preserve digital object over time, and we will see later that it could be a lot of time. Also that they have the features to maintain the data integrity and authenticity. They should demonstrate or be transparent in providing information about sustainable governance and funding. If it's needed, ensure secure and controlled access, and of course have enough documentations to describe policies, workflows, and to identify responsibilities on the different aspects of the repository. So this concept was discussed in June 2022 by a group of some European repository owners, and they realized that should be some network of repositories to ensure somehow that they can support the fair data and they can trust each other and providing trust to the researchers in publishing the data. So coming from this discussion and a paper, the FIDELIS project was created. It started last year, and it aims at develop a network of repositories that can self-supervise their features in terms of fair data and trustworthiness. So the objective of this project that CSIC is participating in is to set up this European network of trustworthy repositories, but we will not only focus in European, but we will open to more countries that will support the development and growth of trustworthy digital repositories within, in particular, the EOSC ecosystem. Well, one thing is to improve the interoperability across repositories, because the idea is to facilitate the reuse and the integration of the data. So it will be focused on how can interoperability be improved, and to strengthen the upskilling of repositories. This will be done thanks to different support training that will well help repository administrators and etc. to improve their feature and to their skills. In parallel, we have another project that is sister of Fidelis, this EOSC Eden, and it is focused more on the curation and preservation part. So for be trustworthy, the data repository need to secure and ensure the integrity of the data, but also it should help to curate the data and to preserve the data in a long term. So this project is more focused on developing different tools and solutions for curate and preserve the data. Both projects have similar objectives, let's say, but they are different. Both of them they actually have created different networks that the target is basically the same. Fidelis has established a network of trustworthy data repositories, but Eden is establishing a network of curation experts. Okay, so they can potentially help to provide some training to the Fidelis network. In the case of standards, Fidelis will recommend some standards, APIs, and solutions in general to foster interoperability. And it then will be focused more on curation, even creating new tools for facilitate the curation and long-term preservation of the data. And so this is more on that part of the data lifecycle. Regarding the EOSC Federation, Fidelis will facilitate the technical federation of repositories, and it then will be a registry of tools that could help to that network of trustworthy repositories. You can find more information about this in the webpage of both projects that is integrated. Okay. Regarding trustworthy data repositories, so far we have a certification that is called Trust Seal that evaluates whether a repository can be considered trustworthy. And it is be done assessing a set of 16 requirements that cover different aspects on the repository, both at technical level, but also at governance and operation level. For example, this certification checks that the repository can be sustainable. This means that they have enough long-term funding because they are structural of an institution, or they are supported by a bigger entity. This certification also checks the data management and curation practices, that checking that everything is well documented and every detail is accessible. It checks the policies, for example, in preservation, that should guarantee the long-term authenticity and usability of the object, even migrating the formats if needed because of obsolescence, and different details about the infrastructure where they are deployed, and also security issues like, for example, backup procedures, protection of sensitive data, etc. This is quite complex to obtain this certification. So basically, one of the alternatives that Fidelis provide is try to create a way to self-assess the level of trustworthiness. So in Fidelis, we have created the TTRAM, that is a matrix where you can exactly check the trustworthy of your repository thanks to different attributes. And the idea is that it will not be a certification, but a kind of model that helps repositories to identify some strengths, some gaps, and some areas where the repository need to have some upskilling. Okay. So it checks also some governance and organizational maturity, like policies, roles, etc. Different details about the technical features, like the level of interoperability, the APIs that they provide, the scalability and system robustness. Also, it's very focused on fair data practices, checking how repositories can be fair. Regarding security and access control, also some self questions about how this is managed, and also, well, some services of support for the users. So the idea is that you have this matrix with some information that can allows you to evaluate the level of compliance for different aspects in your repository. As I said, at technical level, but also at governmental and policy level. The TTRAM allows you to collect the information in a logical way, understand the different best practices and solutions that you provide, and also helps you to share that information to someone else that can, well, help you to identify some potential improvement. And it is very useful because it allows repositories to identify exactly where they need to be focused on get more training, for example. As I said, one important thing on this type of repositories is to provide fair data. I guess most of you know what are the FAIR principles. Whenever we talk about sensitive information, there are some tensions between this fair that, well, fair data does not necessarily mean open data, but some of the indicators and the principles are very close to open data. So sensitive data can still be fair if the metadata is openly available, for example, if the access conditions are very good defined, and if the authentication and authorization mechanisms exist. So within this relationship among sensitive data and FAIR principles, we have released last year our deliverable for World Package 5, where we identified within the list of FAIR principles different aspects that implies tension. So you want to get more information, you can check that document where everything is defined. So whenever we have anonymous data or even sensitive data or open data, we can evaluate and assess the level of fairness. Okay, there are different tools that allows you to do that, but in this case, we are going to present FAIREVA. That is basically an assessment tool to check research outputs fairness level. Okay. We started from the list of indicators of the Research Data Alliance that, well, details the different principles. And we aim at get some modular, scalable, and flexible system to allow you to develop your plugin for your specific repository or data portal. FIDELIS is not a service, so probably you won't be able to find a globally working instance with FIDELIS. but you will find some different instance. But it's a generic tool that you can customize for your specific case. As I said, the starting point is the RDA indicators that are 41 specific checks to get more details about the FAIR principles. But it is quite generic, so it is applicable to different domains. And the idea of FAIR is to provide an architecture where you can define your own plugin to connect to your system, okay? Because probably you will have different metadata schemas to define your research outputs. You will need to check specific details about that schema and the types of data that you have within your system. So it is important to not only having a generic way to do that, but also to specify for your specific system the type of test that you need to do. Okay, the typical test that we are doing is something like that. For example, to check fairness, we check if you have a persistent identifier assigned to your digital object, and we check the type of that. We can check regarding accessibility if the metadata can be accessed through a standardized protocol, like for example, OIPMH. Interoperability, we check different control vocabularies, and for reusability, we can check details about the license that you have assigned to your data. I'm going to show a very brief demo on how to use FAIR EVA. And so let me share my console. You can find the Dockerized version of FAIR EVA in this GitHub repository, where we have a Docker Compose where you can simply get the system up with two components. The FAIR EVA, that is the API where basically the engine is run and the tests are performed, and the web interface. Whenever everything is up and running, we can launch on a local host in this case, and we can just put here the identifier of the digital object that we want to access, and we can select the plugin. In this case, we have two plugins installed, OAPMH and GBIF, and we are going to access one digital object from GBIF. We can check run test. In the meantime, just to let you know that you can configure within this repository the list of plugins that you want to install on your instance in the config file, and it will install you the different plugins that you need. So whenever everything has run, we will obtain different metrics about the different indicators that the RDA propose in findable, accessible, interpretable, and reusable. You can get information about the different scores and some tips about how to improve this score. And one interesting feature of FIDELIS is that you can add more specific data tests. For example, in this case, we check the quality of data in biodiversity with these metrics, but you can develop your own system to check quality. One of the potential things that we want to check is that if we could add some anonymization checks within FAIR to get some results about the level of anonymization of your data. That could be interesting. So that's all from my side. So if you have any question, I will be glad to answer.
Thank you so much, Fernando. There are a couple of questions to you. So one is how fair metrics can be evaluated in a data set. For example, how can you evaluate the interoperability with any kind of metrics?
It depends on the type of indicator. In the case of FEREBA, we are adopting the RDA indicators that are 41. For interoperability, there are like 12, 15 indicators, and the type of things that we check is that if in your metadata schema you are including some control vocabularies that allows you to identify univocally some information. And if you are using metadata standards, that helps to improve the interoperability. So you can check the content of your metadata, but also the type of metadata that you have used to improve the interoperability.
Okay, thank you, Fernando. There is another question in how does FEREBA compare to FairChecker or Fuji?
Well, the main difference, FairChecker I don't know a lot, but Fuji I do. So Fuji is kind of generic. I think FairChecker is also. So you can give a DOI or a persistent identifier, and it will allows you to get some scores, but it is quite generic. So it is, well, it will give you some information at general level. The main difference of FEREBA is that you can develop your own plugin and you can implement exactly how to get more information that can be very rich for your data system and to get the list of features that you are providing and the type of metadata schema and type of data that you are using within your community or within your own repository. So that's the main difference.
So it would be customization, I would say.
Yeah.
Okay, and then there is another question. Will FAIR-EVA evaluation results be integrated into Red3 data repository?
Red3 data, I think, is more for repository themselves. So since FEREBA is just a tool, it will provide you some metrics. But I'm not sure, but at least in Fidelis, in the TTRAN, one of the things that they check is that if you provide any kind of fair assessment within your repository. In this case, you can mention, if it is the case, that you are using FAIR-EVA to provide information to the users about the level of fairness. But it's like different categories of services. But you can, yes, use FAIR-EVA to assess the fair data on your repository.
Okay, thank you, Fernando. Then we have two final questions that are more broad, and with that we can conclude. So one is, what is the role of Siesta and the EOS concerning data repository, data publishing? Sorry. So in Siesta we are working on a data analytics tool that allows you to consume sensitive data for your data analysis. In that regard, we are also working on the data release tools that will allow you to check if will allow a data right holder if a given derived data is able to be published according to the security restrictions they want to imply. And with that, we will work on connectors with existing repositories, but we will not provide within Siesta, let's say, a sensitive data repository on ourselves. So this is our positioning. And then the last question is about the presentations. Yes, we will share the presentations. We will upload them both to Zenodo and to the Indico page that will be linked in this webinar, in this YouTube video, so that they are available for further reference. So with this, we can conclude our webinar almost in time. So let me thank again the speakers for your availability today. Thank you to the audience for joining us and for participating in the debate via the questions. I hope you have found our webinars interesting, and let's be in touch for additional sessions around sensitive data. Thank you, everybody, and bye-bye.